Hi, everyone. Welcome back to BIEB 152, Evolution of Infectious Disease. This is lecture number 11, and today what I'm going to do is review the material from lectures 1 through 8. That material is going to be on the midterm, and so I will go over the subjects that I think were really critical to understanding this material. There will certainly be material on the exam that I do not cover in this lecture, but is a part of the slides and the previous lectures. So one of my goals for this lecture is not just to review the material, but to go over some subjects that I've had lots of questions about so I can help clarify those subjects. And the other goal is to make connections between the different lectures. I try to do that within the lectures, but I don't always have time to do that. And so this time I have the opportunity to make these connections. And so hopefully by building different connections in your brain, it'll help you remember this material a little bit better. It'll be more interesting, and it'll show you sort of how the class is building. And then when we go into the next phase of the class, you'll be learning more specific ways of analyzing modern diseases, and also just more facts about those diseases and how those diseases evolve. Checking the temperature on COVID-19. Okay, you guys have seen this. You've seen this graph many times. This is a new one, I think, from yesterday. And the question is that it feels like we all have this sort of psychological idea that we would just have to shelter in place for April, and then May would come and summer was coming, and that we would all be able to sort of reopen our governments and reopen our states and reopen the country. And so cell phone data is suggesting that people are already beginning to relax their shelter in place and are beginning to travel much more than they were when the shutdown first happened. And then the question is whether or not this is appropriate. Should we be reopening our government? And of course, this decision should always be based on data. And so if we look at the data for the entire United States, we can see that our actions of sheltering in place have stopped the exponential growth of COVID-19, and that we've bent the curve and we've caused it to plateau. But we haven't caused it to actually drop in its rate of spread through time. It's basically stable at about 30,000 new cases per time. There does look like there's a little bit sloping downwards of that trend, but overall it seems that we just have a consistent level of spread of COVID-19. So just looking at this data, I would say that I don't know if we should change our behaviors. Certainly if we do change our behaviors and we begin to go to stores again and things like that, we must alter the way that we used to go to stores or go out in ways that continue to effectively social distance, maybe without us actually being isolated in our houses. So I am a little bit nervous that we'll have another uptick. Of course, remember, we had this theory that perhaps the disease will be a seasonal disease and won't spread as well in the summer. And so it might be the case that we open things back up and the disease is unable to spread because it's so hot, it's sensitive to UV light, and it's sensitive to humidity. Now that's a huge assumption, and I guess I would like to see better data on whether or not this disease is going to be seasonal before taking the risk of triggering a second rise in the disease. And so, as I've said before, I don't think it's great to look at the United States as a whole and look at these numbers as a whole. We really do have to think much more locally. And so if we look at the data for individual states, there are, and this is also from the New York Times, sorry I didn't cite it here, but you can see that there's kind of three behaviors in the different states. And so there's many states in which, and so that's this top behavior up here, in which the rate at which the disease is spreading is continuing to increase. And so if you're in one of those states, that you don't want to change your behavior in the way of opening up and facilitating more human-to-human interactions, you want to change your behavior in a way to dampen human-to-human interactions and dampen the spread of the disease. And so those states, it's not advised to reopen, especially it doesn't seem like it has had too much of an effect, or it had an effect and they've changed their behavior and now the disease is beginning to increase again. That's the case for Vermont and Alaska. So we in California are in this other category, which is, it looks like we've changed our behaviors and now we kind of have more of a plateau pattern. And so this is a case where my question is, why stop doing a good thing? Obviously, we slowed things down, we slowed the spread of the disease, but we didn't drive it out of our states, and it's still spreading relatively fast, spreading, in most of the states as fast as it was on the highest day in which it spread the most. And so I would say there, I don't know, I'd be a little bit more hesitant. Now, certainly what Gavin Newsom, the governor, has released yesterday for Friday, that some stores are going to be open, but that's for curbside pickup. And there's some other things that will be relaxed, such as people are allowed to work in offices that can't work remotely from home, some other things. And so those are pretty minor. I don't know if that's going to trigger an uptick. And it seems just that people are on their own beginning to come out of their houses more and interact a little bit more. So hopefully we don't cause another uptick there. But California, we have been pretty cautious. And so I'm pretty confident in that if we follow the state guidelines, nothing too bad will happen. And then the other states, there's another category, and this is where the numbers of cases are really declining. And it's very promising. And I think in these states, give it a shot, open up, but do it with proper surveillance. Have a lot of tests so that when if the disease starts to spread, you'll be able to detect it quickly and shelter in place again. Remember, many lectures ago, we talked about that sort of up and down spiking of the disease and how to maintain it at low enough levels so that we never overwhelm our healthcare system. But what that required was constant surveillance and responding to when the disease begins to spread by sheltering in place again. The one example on the screen that I think is really interesting is New York. New York was crazy. Tons and tons and tons of people were infected. And it turns out that they are now doing a lot of testing for antibodies. And there are some problems with these tests, but at a population level, they do give you a read on how prevalent the disease actually is. And it looks like somewhere between 10 and 20% of New York. So that's an extraordinary number of people, not just New York City, but upstate New York as well, have experienced COVID-19 and are recovered. What that means is that they're beginning to approach a level in which it's going to be harder for the disease to spread just because there's not as many sensitive people to the disease. And so they're not at the point yet where they have what's called herd immunity. We'll go over the math of herd immunity later in the term, but they're not immune to the disease from spreading as a population yet. That's called herd immunity, but it does mean that it's going to have a harder time. And I suspect that this drop is caused by behavior, but also probably even more so because so many people are immune to the disease in New York. So that's pretty interesting. That number of between 10 and 20% also jives with what we know about how many people have died from COVID-19 in New York. And if we know that there's a 1% death mortality rate for COVID-19, that would mean that for every person that died with COVID-19, times that number by 100, and that should give you the total population of people that actually do have COVID-19. So that calculation actually makes sense with this 10 to 20% number. And so I am pretty confident in those studies. Okay, so I don't know, there's a mixture of responses of the disease. It's a patchwork. We're a patchwork country, and it's a patchwork and how it's affecting different states at different times. And so we do have to think locally and focus on the dynamics in our communities and be responsive to any increases in the disease. I always like to give a little good news about the technology development in treating COVID-19. And it does appear that there is maybe another strategy that we haven't talked too much about. It's a human monoclonal antibody. And so basically we talked about that plasma technique of taking out the plasma from a patient that has recovered from COVID-19 and giving it to a patient that is experiencing a bad case of COVID-19. And this is conceptually very similar. What you're delivering in that plasma is are antibodies. And what this is being able to identify a human antibody, a single one. That's why it's called monoclonal. And you can even produce these in the lab, and then you can administer them as a therapy. And it turns out that this one that they've identified, so the name of it is 47D11. It is obviously, it's having effects on both reducing the infection caused by SARS-CoV, so that's the original SARS, and SARS-CoV-2. So it seems like it's a very generic antibody that will be effective at suppressing SARS-CoV-2, but also other coronaviruses as well. So that's encouraging. That's also encouraging because if it has this sort of broad spectrum in ability to combat disease, then it must be targeting something that's very generic and general to these viruses. And those features tend to be conserved in evolution. And so it would be unlikely for SARS-CoV-2 to be able to evolve to avoid this antibody. Of course, evolution is tricky. Maybe they would be able to, but this is really hopeful news. So of course, there's lots more development that has to be done on this therapy, but it just shows you another angle in which people are trying to develop effective therapies. We have, thousands of labs all around the world developing all kinds of new strategies. And so I am hopeful that the disease will be seasonal and we'll have a relief in the summer. And hopefully by the fall, we'll actually have some therapies in place. And then by the winter, maybe even have a vaccine. That's obviously very hopeful, but I think as a species, we can do it. We certainly should strive for it. And of course, remember, we have remdesivir. It has some effective ability at reducing the time that people have the disease. So that's also good news. Okay, so now let's get to the actual lecture, this midway review. Let's start from the very beginning and think about just the evolutionary process and how it works. So we have a population of bacterial cells. They are the population size we call N, that's the variable. And these bacteria are all identical to each other at first. And as they grow and they divide, eventually one of the bacteria is going to get one single mutation in one base of its DNA. And so as the population grows more and more, there'll be more and more mutations. So the time that it takes for a population to go from N to 2N is called the doubling time. That's this TD variable listed on the screen. And that is literally just the time that it takes to from one cell to turn into two cells, or for a population of N to turn into a population of 2N. This doubling time is directly related to the growth rate that we talked about. So the growth rate is R. The growth rate is given in this exponential growth equation here. And the doubling time is directly related, as in it is ln2, so that's just a number, divided by R. So it's directly related, but remember, and it's kind of fun to think about sometimes, it's inversely related. So and this makes a lot of sense, as the growth rate becomes faster and faster and faster, the time it takes a cell to go from 1 to 2 cells will shrink down lower and lower and lower and lower. So just remember that they are directly related, but in a reverse, inverse fashion. So these mutations will accumulate, and they accumulate at a very predictable rate. So once you know a mutation rate, then it's easy to predict, as a population is growing, roughly how many mutations will occur in that population. Remember, mutations are random, so this is just an estimate. It's a kind of average behavior, but we can be pretty good at predicting roughly how much genetic variation would be in a population. And so we talked about three types of mutation rates. There's the mutation rate per genome replication, that's mu-G. So that's just the rate at which you get one mutation in an entire genome. And then there's the per base mutation rate. That is looking at just a single base in the genome, what's the rate at which that base will change as the cells are dividing. And so the mutation rates, the per base and the per genome mutation rates, are directly related to each other in that the per base mutation rate is going to be the per genome mutation rate divided by the number of nucleotides in the genome. So when you're answering these mutation rate questions, always ask yourself whether or not you can find the per base mutation rate. And that's sort of the baseline mutation rate. And then you can calculate, what's the mutation rate given a gene of 1,000 bases, or what's the mutation rate given a genome of 1,000,000 bases, or so forth. So always break it down to the mu-B and then build it back up to say, what's the frame that I'm looking at? How large is that frame? If it's 1,000 nucleotides, then I just take that per base mutation rate times 1,000, and that'll give me the rate at which I expect to see accumulation of mutations in that gene. And we actually talked about a really interesting experiment, this Luria and Delbruck experiment, that proved that mutations were spontaneous and were not induced by the environment. And it also, in the same study, they gave us the math to translate the data from their experiment into calculating a mutation rate. And this is the zero method that I talked about with you guys. And so here's the equation for the zero method. And you actually combine these two equations together. So you find M first, and then you can solve for mu R. Okay, so there's a lot going on the screen. So let's just sort of walk through one bit at a time. Mu R is the rate of mutations, it's a per genome rate of mutation, but it's not of any site in the genome that can mutate. It is focusing on the number of sites in the genome that confer resistance to a phage, or maybe resistance to an antibiotic. And so mu R is the per genome mutation rate, but of this phenotype that is resistance to something like an antibiotic or a phage. So this is our way to calculate mutation rate, and we can then compare mutation rates across different species or so forth, or to different antibiotics. But unless we know the number of sites in the genome that, if they mutate, can give you resistance to the drug or the phage, then we don't really know. We can't convert the mu R into mu B and then into a mu G. But if we do know the number of sites, so say there's 10 sites in the genome that if they change, they give you mu R, then you can easily convert mu R into a per base mutation rate. And then you can convert that per base mutation rate into a per genome mutation rate if you know the number of bases in the total genome. Do make sure that you understand how to convert from mu R to mu B to mu G. And so this is just a list of what all these variables are. You want to solve for M. That's what you don't know. You know N, that's the number of cells in the test tube at the end of the experiment. So that's basically the number of opportunities where a mistake could have occurred and a mutation could have occurred. And then P0 is just the number of plates that have no colonies on them at all. It's the proportion, the fraction. Then mu r is the per genome replication rate for resistance mutations. And so you can just plug and chug and solve the problem. So basically, you can calculate the average number of mutations that occurred in these test tubes. But if you want to then get that number and make it more relevant, turn it into a per genome mutation rate. That's why you divide by n. That's the number of genomes that were in the test tube, and so that gives you the per genome mutation rate. Okay, so we get a mutation. This is I'm just showing you just sort of the how I'm going to be using these different symbols through the next set of slides, where this is the ancestral DNA, and here right in the middle of the sequence there is a G has turned into an A. Okay. So some mutations actually have very little to no effect at all on the phenotype. And so these mutations are affected strongly by this process called genetic drift. So evolution isn't just about natural selection. There's other evolutionary processes that also drive changes in frequencies of genes through time. And one of these processes is genetic drift. So genetic drift acts most efficiently when mutations don't have any effect on the phenotype and are considered neutral. But genetic drift also does influence the dynamics of mutations that are beneficial or are deleterious. And in previous lectures, we talked about how in very small populations, genetic drift takes over, but in very large populations, natural selection will take over. But both of these things are happening all the time on every single mutation. It just matters less. Genetic drift matters less if the mutation has a beneficial or deleterious effect. Okay, so let's focus just on genetic drift for now. And so it's sort of a random process. We talked about flipping a coin. We went through that whole procedure where I was flipping the coin and replicating cells on the whiteboard. And so picture that, remember that demonstration, and think about how this is a random procedure. And so here's just some text that goes over that. Imagine that each generation the cells divide, they double their numbers, but the environment can only support the initial number of cells, so half of these cells are randomly removed. Sometimes we randomly remove a disproportionate amount of the mutant or non-mutant. Randomly, the mutant can increase in frequency and fix, or decrease and drop out of the population. So that's what these images of these populations through time are trying to describe early dynamics in this scenario or that scenario, where you have the mutant, even though it's not beneficial, increasing in frequency through time and eventually fixing. Here, the mutant is not deleterious, but it comes into the population and then drops back out of the population for random reasons. If this is confusing, there are actually a ton of YouTube videos. So if you didn't follow on the board when I was doing that, definitely go onto YouTube, type in neutral genetic drift, and there'll be a ton of videos about it. So check those out. Okay, so evolution, our goal in this class is to predict evolution, to understand the processes so that we can predict whether or not diseases will evolve, how they'll evolve, whether or not that evolution is bad for us, and if it is bad for us, how we can actually stop it. And so right now, at this point in the lecture and in the course, I've told you about random mutations, and now I'm telling you about random genetic drift. And so how do you predict a process that's so contingent on randomness? Well, there are behaviors, just like there were predictable behaviors about related to predicting genetic variation caused by a certain mutation rate and a certain amount of growth in the population of cells. For genetic drift, there's also other predictable behaviors. So for genetic drift, we know that drift is stronger when the population size is smaller. And the way that you can understand this is that it's a coin flip process, right? And so if you only flip a coin 3 times, there is some chance that you would get 3 heads. It's not unimaginable that that would happen. But if you flip that coin 30 times, it becomes nearly unimaginable that you would get 30 heads in a row. And so that's basically the same idea with genetic drift, that there's more stochasticity in the outcome of evolution in small populations than in large populations. And so, of course, if you have an infinitely large population, that's the scenario where drift goes away completely. But if you don't have an infinitely large population, i.e. every population that exists, then there's some level of stochasticity and some level of drift. We talked about in class how, if you are given a mutation that has a 1% fitness benefit in a haploid population, that once you get to a population size of about 1,000 or more, then drift really has little effect on determining whether or not that mutation fixes in the population. And so that's kind of the cutoff threshold where you really have to focus on selection more than drift. But drift is always happening at any population size. It has just a smaller effect at these larger population sizes. So one of the predictable things that we know about drift is that the frequency of a given allele is its probability to fix in the population. Therefore, 1 minus the frequency is the probability that it'll go extinct in the population. And from that, you can easily derive that a new mutation, which is just one individual in the population gaining a mutation, is 1 divided by the total population size, gives you the frequency, or the probability that it'll fix in that population. And so obviously that probability is low, when you're just a single mutation, and the probability of that new mutation of going extinct is pretty high. But it's a function of the total population size of the population. And so that 1 over n is just frequency. It's a way of describing the frequency of a brand new mutation. A brand new mutation, by definition, is just in 1 genome in the entire population. So this is demonstrating the simulations that I ran for a population that was size 20, and showing that in just 1 out of 20 simulations, we actually saw the neutral allele fix in the population. So this is consistent with that theory. You could have 20 simulations where none of the mutations fix. You could have 20 simulations where multiple mutations fix. It's just this one, this is a probability, and so this is the sort of average behavior, but it's not definite because it's still based on a stochastic process. You can see, so this is generation on the x-axis, and on the y-axis, these are just different individuals, so it's not really an axis. These are just 20 individuals in the population that replicate. Some of them do not replicate. Other ones replicate multiple times. And so from one generation to the next, you have this stochasticity and this drift of the mutant allele. And in this population, you can see the red allele has spread and fixed. So another characteristic behavior of neutral genetic drift is this neutral mutation rate of neutral mutation substitutions. And it is directly equal to the mutation rate. So I went over this a lot in class. I think it's really elegant, and I really like this conclusion. And it's important for understanding because it's the backbone of why we trust things like molecular clocks. We talked about molecular clocks. They won't be on the exam for the midterm. They will be on the final. We talked about them 2 lectures ago. So, and it's this really nice thing where the equation that we would write in order to predict the neutral mutation or rate of neutral mutation substitutions is right here. So wait, let me just describe first what is rate of neutral mutation substitution. So say you're given a gene and has 1,000 base pairs in it. What I'm asking you is, what's the rate at which that gene, that frame that you're looking at in the genome, would accumulate mutations through time? And so that gene is going to mutate often as cells are dividing and during multiple generations of evolution. But many of those mutations will actually just be lost randomly from the population. Actually, the majority of those mutations will be lost. And so what a substitution rate is, it's not a mutation rate, it's the rate at which a sequence across many, 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 many generations will accumulate genetic differences. And so that rate is determined by two different processes. Of course, the first process is the influx of new mutations. And so that's what I'm showing you here are two different populations of two different sizes. And we walked through this a lot and did the math behind it in the drift lecture. But what I'm showing is that there's a certain rate at which you get new mutations. And that is a product of the per base mutation rate times the genome size, and then also times the number of genomes in the population, the N. And so that is this mutation rate. And so that is determining how many mutations are influxing into the population. And then, but that's not determining the substitution rate. You have to, if you're going to substitute a site in that gene, that rare mutation now has to spread in the population and fix. When that fixation happens, that's when substitution happens. And so there is all of these mutations, most of them will actually just come into being and then drop back out of the population for random reasons. But, 1 divided by the total population size, so for here, 1 in 10, will spread through the population, randomly fluctuating until it fixes in that population. And so that is when you count it as a neutral substitution. And so the rate of neutral substitution is going to be a product of the population size times the underlying mutation rate. This is a per genome mutation rate, or if you're looking at just a single gene, per gene mutation rate. So that's why I haven't designated what exactly the B or the G or whatever is here. You have to sort of alter it by the frame at which you're looking at in the genome. So total population size times the mutation rate. And then this is the influx. This is the red ticks on the graph. And then this is the chance that any of those mutations actually fixes. That combines together to give you the rate at which you get this event happening. That's a neutral substitution. And what is really nice is that N is in the new denominator and is also in the numerator. And so this simplifies out to just the mutation rate. And so mutation rate will be mu B times the number of nucleotides that you're studying. Okay, so I really love that. It's nice that it simplifies down. And so we have a reliable way to actually clock how long ago two different organisms diverged from each other just by looking at their modern sequences and counting up the number of differences. One other predictable feature of neutral genetic drift is that if a mutation does fix, so most neutral mutations don't fix, they drop out of the population. But if a mutation does fix, it is on average going to take 2 NE generations. And so this is something that we went over thoroughly in the homework. So make sure to reevaluate that. But what I'm showing you here are 10 different population genetic simulations for a population size of 10. And so we see that 1 in 10 did fix in the population. So that's consistent with our predictions that 1 divided by N is the probability that a mutation will fix. So N here is 10, so 1 in 10, 10 simulations. So, but the other thing that is in line with our theory is that it's taken about 20 generations to actually fix in this population. That's not a hard rule, that's just an average behavior. But it is, if you look at enough simulations, you will find that it is 2NE, the average time it takes for a new mutation to go from one mutation to fix in the population. So population size matters for neutral genetic drift. And so how do you calculate population size if the population is fluctuating up and down, up and down, up and down? And we went over this, this different way of calculating the average, the harmonic mean. And what this does is shows you that the bottlenecks, the low population sizes, have a disproportionate effect on the effective population size. So this idea, this is a new variable, NE, is the effective population size. So NE will equal N if there's no population fluctuations. But if it does fluctuate, then you have to use this harmonic mean to calculate what the effective population size is. Okay, so that's the case if that new mutation is neutral. But what if that new mutation is beneficial or deleterious? What happens if it actually changes that amino acid in a way that affects the fitness of the organism? And so there's, obviously two possibilities if it's affecting fitness. One is that it's improving the fitness of the organism. And so what will happen there is that mutation will relatively rapidly fix in the population in a much more deterministic way than mutations that fix due to neutral genetic drift. If that mutation is deleterious, it will actually really rapidly drop out of the population in a much more deterministic and faster way than due to neutral genetic drift. So mutations have kind of a spectrum of different effects on fitness. Remember, fitness is basically how fast a mutant grows compared to how fast the wild type grows, or how fast the evolved genotype grows compared to its ancestor. And so this is just a hypothetical distribution of mutational effects. So this is just a histogram. This is the number of mutations that have different effects on fitness. And so we would hypothesize that there'd be a glut of mutations that have no effect at all on proteins, that there'd be some small number of mutations that would improve the organism's fitness. We think most biological systems have been evolving for a very long time, and so they're probably fairly optimal. And so there's few ways for biological systems to improve above what they've already optimized. And so that's why we think that this is there's relatively few beneficial mutations. There's lots of deleterious mutations, and then there's a lot of mutations that completely screw up the cell or screw up the virus in a way that causes it not to be able to replicate. And so that's obviously a dead end once you get that kind of mutation. So this is not just theory, we can actually measure it. We've measured it in lots of different organisms. Now, this is one of the first. This is in a virus, so it's relevant for this course. Yeah, it shows you that the distribution is very much like we thought. Lots of ways to completely destroy the replication of the virus, an influx of neutral mutations here, and then just a small subset of the mutations are actually beneficial. Okay, so if we do have a beneficial mutation, can we make predictions? So first, can we measure how beneficial that mutation is? And then can we use that measurement of its benefit in order to predict how fast it'll spread through the population and how fast it'll even fix in the population? Or we have an equation where we saw that we could say in 10 generations in the future, how common do we think this mutation will be? What frequency will it be at? And so this is our measurement, our way of estimating Darwinian fitness. This is W, and this is W for the evolved. And so we have the growth rate of the evolved divided by the growth rate of the ancestor. And what is growth rate? Well, this is an equation that we derived from that exponential growth equation, and that growth rate R is equal to ln, the number of cells at the time point at the end of the experiment. Remember, we're running these competition experiments in order to measure R, measure relative R, and be able to estimate fitness. And so we measure the number of cells of a given type at time point 1, number of cells at a given time point, cells at a given time point, or time point 0, and then we divide that by time. Remember, one of the shortcuts to calculating this is that time, because these experiments are being run as a competitive experiment where the cells are growing for the same amount of time, time then gets divided out. It's the same in the numerator and the denominator. And so really you just have to calculate the numerator up here for the R's and not worry about time. Okay, so that's how we calculate relative fitness based on different exponential growth rates. And then, we had all of these different equations. So this is the exponential growth equation where we find R. Then once we find R, we can calculate the doubling time. And then when we have the doubling time, that really gives us a parameter for how fast these things are growing. And then we can plug that doubling time into this larger equation, which predicts the frequency of the evolved allele at some time point in the future based on where it is right now and relative to how well it grows, so its doubling time compared to its doubling time of the ancestor. So this is making prediction if you just have two types growing with one another, you have the ancestor and the evolved type, you can predict how abundant that evolved type will be in the future. So one of the things that I had brought up early in the lecture about natural selection, which I did not have the chance to really explain very well and to get back to later in the lecture, is that in this course we're going to talk about two qualitatively different types of beneficial mutations. And I just want you to sort of be able to sort of understand why they're different and to square that so that you don't get confused sort of later on in the lecture. So what we're talking about here with this relative fitness, I said before, is really you can observe on this massive petri dish where genetic mutants of bacteria are competing with each other for space and resources on this giant petri dish. The giant petri dish also has a gradient of antibiotics. And so the ancestor isn't able to move into this section of the plate until it gets a resistance mutation. And so I had said before that, what we're calculating is basically the relative size of these fans. And so the red is doing really well. That mutant there can grow really well in this antibiotic environment, whereas this blue mutant can grow there, but it's not doing nearly as well as the red. And certainly the yellow is not doing as well, and the orange aren't doing as well. And so if you're looking at this equation and you're sort of wondering to yourself, like, well, how can I visualize this? This is how you can visualize it. You can look at these shapes that I've taken from the plate, these what I would call fan sizes. They don't quite look exactly like a fan, but you can see what I'm talking about. And really, what you're doing in this calculation is seeing sort of how much this genotype has grown and how much this genotype has grown, and you're just making you're taking the log. That's just because things grow exponentially, but then you're comparing how much that growth has happened relative, so the red relative to the blue. And so really you're comparing sort of this log transformed area versus this log transformed area, and that's what's giving you the relative fitness. So it's really simple to see sort of how that equation relates to these bacteria growing, some growing faster than each other and pushing each other out of the petri plate and eventually out of the population. But there's this other kind of adaptive mutation that's happening where mutations are actually giving you the ability to go into a higher dosage of the drug and then another higher dosage and another higher dosage and so on. And this kind of adaptation is very significant. It's actually allowing the bacteria to grow in a completely new environment. So it gets released from competing with its ancestor because it is able to go into a space that the ancestor can't even grow in. And so if you use this equation to describe what's happening here, the equation actually breaks. And so it really is it's a quantitatively and a qualitatively different type of natural selection. So what's happening here is that, so say we have fitness of the resistance, and we're comparing the fitness of the resistance compared to the sensitive, and we're measuring this fitness in the presence of the drug. And so in the presence of the drug, the resistant one, it's going to grow. So you're going to have a high number here and a low number here. But down here, this guy is unable to grow in the presence of the antibiotic. And so say this is a bacterial static antibiotic, and you have one cell here in the initial, well, you'll have one cell at the final as well. So if the initial minus the initial divided by the final, if the final divided by the initial are the exact same, you'll get a value of 1. So ln1 actually equals 0. So it just breaks this equation down. And so you can see mathematically why this type of adaptation is different. But you can also sort of see just more kind of holistically that this is, gaining the ability to grow in a space where the ancestor was unable to grow. So this is all you really have to do in order to predict the spread of the antibiotic resistant type relative to the ancestor is just consider how fast the antibiotic resistant type is growing, because that ancestor is no longer a competitor. It's either killed by the antibiotic or unable to grow. So I hope that makes sense, how these two types of selection are different. Some people call them soft selection and hard selection, but that's used in different ways throughout the literature. So I would just sort of focus on competing for resources versus gaining antibiotic resistance and gaining the ability to inhabit new spaces. So what we learned is that benefit of a mutation, even a resistance mutation, is often contingent on the genome that occurs in. Some genomes, those beneficial mutations will have large effects. Other genomes, it could actually even have deleterious effects. And so this is a concept called epistasis, and it's that there's sort of mutation by mutation interactions in genomes, and that they affect how beneficial or whether or not a mutation is actually beneficial. And so we talked about this concept of the fitness landscape, and I just want to show you sort of how epistasis would work in this fitness landscape. So say, you have these different points. So these are genotypes of bacteria, for instance, or some kind of organism. They vary in their traits or they vary in their genotypes. And so that's what the x-axis is showing us, is that these are not the same organisms. They have different genomes, they have different phenotypes. And then we have this fitness function that relates this trait or this genetic space to how fit a particular organism will be. And so often we think of these fitness functions as being hills, where this is the most optimal genome that you could possibly get. If you move down, this is suboptimal on either side of that hill. And so say you have a mutation that moves you to the right, adds an extra mutation or gives you some kind of increases the gene expression of an efflux pump or something like that. So that mutation, it moves you in this phenotypic space or in this genetic space on the x-axis over this much. And so if you move over this much and your starting point is here, then you're going to gain a ton of fitness. If you move over this much and your starting point is here, you're going to gain fitness, but not as much. If you move over that much, you overshoot optimality, then you're going to even decline in fitness even more than you already started out at. And so this is not just theory. We have an example in class that we'll get to in a second. The way that you can imagine this is, say this is just one mutation, and you know that you need 4 mutations in order to get optimal drug resistance. And if you overshoot and you have 5 mutations or 6 mutations, then they're going to take you down in fitness. The way that you can understand this, not just as a sort of abstract number of mutations, but maybe you could understand it in terms of mutations that increase the production of efflux pumps. So if you have low production of efflux pumps and you increase the production in the presence of antibiotics, that's going to be really good for you. You're going to be able to survive much better than your ancestor with lower expression. Here, if you already have a high level of expression of efflux pumps, you can maybe increasing it will give you a little bit extra benefit. But there's going to be a point where if you overexpress your efflux pumps, you're going to start creating some other problems in the cell. That efflux pump is maybe going to pump out good things that the cell needs. That efflux pump, maybe there's too many of them on your outer membrane, and it's going to challenge the integrity of the outer membrane and maybe make the cell more susceptible to other things. And so often, there's an optimal level in which you want to express a gene. That would be here for the efflux pump. If you already have too much efflux pump expression that's not really helping you out with the antibiotic and it's causing these pleiotropic side effects, then you don't want to add in more. This mutation now is deleterious and will compromise the cell even more if you get it. So I hope that's a nice, somewhat clear example on how this epistasis works. It's important because it defines how beneficial mutation is, and then that is what we can use to predict evolution. This is that empirical fitness landscape that we measured. I don't want to walk through the details, but the bottom line of this is that there is a fitness peak at 4 mutations, but if you get 5 and 6 and so on, you will overshoot that peak and actually drop down in fitness. So this idea of fitness peaks is not just a conceptual thing, it comes out in the data as well. One example of epistasis that was giving people a hard time in the lecture that I just want to go over again, and it's another example of epistasis that hopefully crystallizes this concept of genetic dependency. This example was compensatory mutations for antibiotic resistance mutations. And we brought this up in the lecture where we were talking about how to combat antibiotic resistance. And I said, a lot of people, or our first strategy to deal with antibiotic resistance is to limit how much we're using antibiotics, because then you won't give bacteria the selective benefit to have that resistance gene. And so it won't spread as quickly in the population. And some people actually have the idea that this should actually lead to the decline of antibiotic resistance genes. And we went over how there's data that actually suggests that even if we stop using antibiotics, the genes for antibiotic resistance are likely to persist for pretty long into the future. And so the problem isn't going to completely go away. And the reason why that is because of compensatory mutations. So I showed you this graph before, and now I just want to give an analogy of what's happening in this graph so that you can hopefully gain a more intuitive understanding for how compensatory mutations work beyond just looking at numbers on a graph. Okay, so let's just start here and then we'll walk through the graph. We're going to start out with wild type. Wild type has a certain fitness. Remember, this is the scenario where we have removed the antibiotic, and so it's no longer in the environment. It's no longer giving the bacteria a benefit for being able to avoid the antibiotic. And so what we're going to compare it to is a car and getting a ding on the side of the car. So I'll walk you through that. So just imagine you have a wild type, you have this car here. You get resistance, and that resistance was to avoid an antibiotic. I haven't thought through this analogy completely yet, so hopefully it works, but maybe you swerved to avoid getting hit. And in doing that, you hit the side of a wall. So you survived, but now your car has a little bit of damage. So you survived the antibiotic, but now your cell has a little bit of damage. So when you get that resistance mutation to avoid the antibiotic, it often interferes with some kind of cellular process. So maybe it's a resistance mutation in the penicillin binding site in the protein, penicillin binding protein. And that allows you to avoid the impact of penicillin on that protein. But now that protein, it was optimized before by natural selection, and now it has a new mutation, and now it's suboptimal. So think of that mutation. It allowed you to avoid this collision, but it had this cost in that it caused a scrape in the side of your car. And so that scrape is this cost. The difference between where fitness started to where fitness was with the mutation is the cost. It's the scrape. So then you can get a second mutation, maybe in that protein or somewhere else in the genome, that repairs that protein. And so it fixes the protein so it no longer has to pay that cost. So that would be the equivalent of painting over this scrape so that you no longer see the scrape, and it looks like it's completely restored to new. Now you know that it does have that scrape underneath the paint, and then the paint has come over and covered it up. So that's resistance and then compensation. But the effect is that it looks like a brand new car still. So why this is an example of a compensatory mutation is that if you were to take the wild type car that doesn't have the scrape in it, and you were to just paint over top the section that should have the scrape, it's going to look exactly the same. You're not going to see any difference in these two different cars. That's the same as with the wild type comparison of the fitness to a mutant that has just the compensatory mutation. They have identical fitnesses. So this benefit that's caused by the compensatory mutation, this difference between the low fitness of the resistant type to the even fitness of the resistant to and compensatory combination compared to wild type, that benefit only is given by the compensatory mutation if resistance is there. There's no benefit to having that compensatory mutation otherwise. It's the equivalent of just painting over top a perfectly well-painted car. Okay, I hope that makes more sense now. Okay, so we've started to talk about antibiotic resistance. So let's sort of review what we've learned about antibiotic resistance. There's lots and lots of different mechanisms for that antibiotics work, and therefore different mechanisms for antibiotic resistance, including efflux pumps, changing the binding site of the protein target of antibiotics, or closing the door on the antibiotics, or changing something about the outer membrane that deflects the antibiotics. So lots of different ways in which bacteria can become resistant. And the way that we measure resistance is by this experiment where we calculate MIC versus MBC. We've gone over this a lot in the class, so I'm not going to go over it too much again and talk about the details of the experiment. But one thing that trips up everybody, which I don't want you guys to get tripped up on, is when you're looking at this row of different concentrations of drugs and different growth of the bacteria, this is growth, this is no growth, your eye immediately goes to where it changes. Okay, so that's what we're looking for, where it changes. And so then the question is, is this the MIC, the minimum inhibitory concentration, or is this the MIC, the minimal inhibitory concentration? And this is the one. And it makes complete sense once you think about it, that this is the level of the drug in which you get inhibition. So this is a minimal inhibitory concentration. This is, a concentration of the drug that's high, but the bacteria can still grow. The same goes for MBC. Remember, this is for how much of the drug inhibits growth, and then this is for how much of the drug is needed to actually kill off the bacteria. Sometimes they're identical to each other for a bacterial cyto or bacteriolytic antibiotic, or sometimes they're separated like this which gives you kind of a hybrid. At some concentrations, it's bactericidal, some concentrations, it's bacteriostatic. So in the next lecture, after we talked about antibiotic resistance, we then moved on and we talked about 6 different strategies to combating the evolution of antibiotic resistance. And so what I want to do here is roughly go over the strategies and the pros and cons. There's other pros of each strategy and other cons of the strategies, but I think that this gives you an idea of, why none of the strategies is perfect. And so that explains why we have so many different ones and how they vary with one another. So strategy number 1, we already went over, slowing the use of antibiotics, hoping that it will slow down the spread of antibiotic resistance genes. And so this strategy is very easy to do. We just have to be prudent with the use of our antibiotics. That is, we have the technology for that. We just stop using them as much. But one of the problems with this is that it won't reverse the rise of antibiotic resistance. It'll just slow down the rise of antibiotic resistance. So that's important. That's our front line. But we have to do other things as well to maintain the therapeutic properties of our antibiotics or develop new antibiotics. Another way is this sort of multi-drug treatment where you're giving 2 antibiotics at the exact same time. This can be effective. It reduces the ability for the bacteria to evolve double resistance. It's harder for the bacteria to do, but you have to make sure that you're using antibiotics that have synergistic interactions with one another. Some antibiotics even have suppressive interactions, and so you certainly want to avoid using them. And some combinations of antibiotics can be toxic to the patient, and so obviously you can't use that combination as well. So that's not foolproof either. Fluctuating drugs, so if you have these combinations that have antagonistic interactions with one another or are toxic to the patient, it doesn't mean that you can't use them in combination. You just don't use them at the same time. You use them at fluctuating at different times. You're oscillating from one antibiotic to the other. And this is also good. It can even reduce antibiotic resistance or slow down the evolution of antibiotic resistance, but only in scenarios where you have collateral sensitivity. If you have cross resistance between the 2 antibiotics, so a single resistance mutation gives you resistance to not just antibiotic A, but also B, then you actually speed up the rate of the evolution of antibiotic resistance. And so that's obviously a problem. So that's not foolproof either. Well, we don't have to just look at antibiotics for new strategies. We can also look at phages and other synthetic biological systems. And so we looked at phage/antibiotic synergistic interaction. This was Paul Turner. And it's a combination of a phage an antibiotic that reduces the phage or the bacteria's ability to evolve resistance to the antibiotic and the phage. And so this is highly effective. One downside of this procedure is that if you find a phage for one species of bacteria or one genotype of bacteria, it's unlikely to work on other genotypes or species of bacteria. And so for each new infection, you might have to develop a brand new phage, and it's like kind of a fishing expedition to find these phages. There's tons of phages in the world. And so you need the ones that have these synergistic properties. And so you have to rely on luck. Certainly you can design efficient ways to screen for phages that have the good property, but still, whether or not that phage is in your screen is still luck. So that's not ideal either. Well, we could actually design phages so that we put in genes that cause them to have synergistic interactions, and we can put in a whole mess of different kinds of genes, and that will give us a huge armory of different ways of attacking bacteria. And so that's a good strategy. This is Tim Liu's strategy. But one of the pitfalls there is that this is a genetically modified organism, and it's much harder. People are afraid of GMOs, probably for unfounded reasons, but there are some concerns about GMOs. And so the FDA, it's much harder to get approval to use a genetically modified organism on a patient. The last one was gene drives, which was this really effective way to actually attack antibiotic resistance genes, replace them with new DNA that wipes out the antibiotic resistance gene, resensitizing the bacteria, and then hitting the bacteria with antibiotics. This is very effective. It was developed at UCSD, Victor Nizet and Ethan Beer. It's in the development phase. It's certainly not approved by the FDA, and it's not in a form yet that could be disseminated into a microbial population or into a patient. But Ethan is taking those steps, and Victor are taking those steps, and hopefully we'll get funding to do that soon. But the question is, people have problems with GMOs. This is a genetic element that actually genetically transforms natural populations of bacteria, transforms them in a way that's helpful to be sensitive to antibiotics, but it's a genetic element that is making these transformations. And so if you have problems with synthetic phages that are genetically modified, then you doubly have problems getting approval for something that is an element that genetically modifies natural populations of bacteria. So obviously there's lots of concerns with that, and there are strategies to dealing with that that are being worked out. Okay, so those are the pros and cons of these different ways to combat antibiotic resistance. If you guys are interested in this stuff, there's other strategies as well. These are the ones that I think give you a nice spectrum of in build up and complexity with how they work. Okay, so in this course, I think one of the most difficult things for students is thinking about biological interactions. And we have a bunch of different interactions, and you have to keep track of them and have them straight in your brain so that you can answer questions on exams, but also just so you understand what's really happening in these, in how these drugs are interacting or genes are interacting or whatever. So biology, like the nature of biological systems is that there's all of these interactions. And I would say that these interactions are both beautiful, but they also really drive you insane keeping track of how they work and keeping track of all the different interactions. So I personally love going out into nature and seeing these really complex ecosystems. But if you then try to understand how those ecosystems work, you have to understand how species A interacts with species B, interacts with species C, and it becomes a big, nasty food web mess, and it's hard to really quantify and hard to really understand. Well, the same is true when we think about a single organism, when we start thinking about its genetic networks and its genes within its genomes, and if we think about therapies and how they're interacting with these different genes in different aspects of the organism's genetic network, it also becomes really complicated. So that complexity, I think, is beautiful, but it also just drives you freaking insane. So with that in mind, what I want to do is just go over a couple interactions that we've gone over in the lab, the class. You can tell that I'm missing working in my lab. So that you just have them straight, and you can see them side by side, visualize them, and sort of then move on and hopefully have a better understanding of how they work. Okay, so the first thing that we were talking about just now are drug-drug physiological interactions. And so that's when you have two drugs being delivered to the cell at the exact same time, and them together will have maybe a really deleterious effect on that cell. And so that's a synergistic interaction, because now those drugs together team up, combine forces, and wipe out the cell. But they can also have a drug-drug interaction where they interfere with each other. And so their effect on the cell is minor. And so that if there's any kind of drug-drug interaction, that's called this physiological interaction. And of course, we have to sort of account for that. So then we have what we call evolutionary interactions. And that is a drug-drug interaction that's facilitated by changes in the genome. So say you have drug A, and the bacteria is exposed to drug A, and it responds in a way that changes its genome, it mutates, so now it's resistant to drug A. Well, that mutation may make it more sensitive, that's collateral sensitivity to drug B, or it may make it more resistant to drug B, that's cross resistance. And so this is it's kind of a drug-drug interaction, but it's modulated through the genome and the changes in the genome that occur to deal with this first drug. And so this is what we call pleiotropy, where you have a mutation that's adaptive to deal with this perturbation, and it has unattended effects on something else. So it can have unattended effects and just make the cell grow slower, or it can make it more sensitive to this antibiotic, or it can also have a dual beneficial effect. It can make it resistant to this antibiotic. And so pleiotropy is this single mutation with multiple phenotypic effects. So resistance to drug A and altered tolerance to drug B. So pleiotropy in general is anytime a mutation has two phenotypic effects. It also happens outside the context of antibiotic resistance. And then the other thing that we've talked about a lot is epistasis. And that's just saying that the effect of mutation A is going to be contingent on whether or not mutation B exists in the genome. And so that can be a positive effect or a negative effect. It's just this mutation by mutation interactions. It's the fact that a specific mutation is going to depend, its effect is going to depend on the larger genome. And so here are all these interactions side by side so you can understand how they relate to each other, but mostly so you can be clear which one is which. And so we have looked at these graphs a ton. Left is physiological interactions, right is evolutionary interactions, describing good and bad drug-drug interactions and good and bad effects of pleiotropic effects of the resistance mutation. We've gone through a lot. I'm going to let you just sort of go back to those other lectures or review this slide, but this certainly will be on the exam. Okay, we're nearing the end, guys. We have two big subjects left. One is this equation on the rate of adaptation. This is a conceptual equation that tells us what kinds of variables we need to know in order to be able to predict the rate of adaptation. How fast will a population improve its fitness over time? This is an equation that's just this conceptual equation that I saw in a talk given by Sally Otto at the University of British Columbia. She is a great teacher and a great scientist, and I really like this equation because it combines a lot of different variables that we've talked about so far in the course. So rate of adaptation is going to be contingent on population size, number of targets of selection, so number of sites in the genome that are good for a bacteria, good for a virus to change, the rate, so the rate at which you get these mutations, and how good are these mutations? That's what this is giving you. On average, how much benefit do these L mutations give you? And so if you combine these things together, you'll be able to tell whether or not a population has a lot of potential to adapt or very little potential to adapt. And so we already did this, but let's walk through how this works. If you increase the population size, then you increase the supply of mutations, not because you're altering the mutation rates, but you're just altering the number of opportunities there are for mutations to occur. And so that same logic occurs with this increasing L. That's the number of targets in the genome that yield adaptive mutations. And so if you have more targets, more opportunities to improve, then you're going to increase the rate of mutations that give you a benefit. You're not changing the underlying rate of mutation, but you are increasing the number of them that yield beneficial mutations. This is very straightforward. By increasing the mutation rates, you'll increase the supply of mutations, and eventually some of those mutations will be beneficial and it'll speed up your adaptation. We looked at data from the Richard Lenski's long-term line experiments that showed us that, in fact, when bacteria evolve higher mutation rates in this experiment, they hit the gas pedal on adaptation as well. Certainly, if your mutation rate got way too high and you started accumulating too many mutations, it would begin to create what we call mutational load and cause the population to collapse, decreasing obviously in fitness. But that is a very high mutation rate that it doesn't tend to evolve. It doesn't tend to happen unless there's something like UV radiation or something like that hitting the population. And so normally mutation rates are relatively low for organisms, and if you increase them, you'll increase your rate of adaptation. The last one is just this idea of the average benefit of a beneficial mutation. And so if you have more mutations in this region of that histogram that we've already gone over, then you're more likely to get a mutation that has a big fitness advantage and accelerate your adaptive evolution. Okay, so the very last subject that I want to go over is about SARS-CoV-2 evolution, treatment, and then maybe its counter-evolution as well. So we looked at phylogenies before of SARS-CoV-2. This is a simplified version of a phylogeny. It's showing you the evolutionary relationship to SARS-CoV and to a couple other coronavirus strains that were isolated in other organisms. So one in pangolin, another one in pangolin, one in bat that's very closely related to SARS-CoV-2. And in this diagram, it's just telling you sort of when different events happened in history. I didn't go over this specifically before, so don't worry about that on the exam. What we did go over was this question of what are genetic characteristics of SARS-CoV-2 that might have helped it jump into the human population. And so now what I want to do is just go back and talk about these things that we've already talked about, but now talk about them with respect to when in the history of the evolution of the strain did these things occur. And so remember, I told you that there were all of these insertions in the S protein. So this is the host recognition protein, and this is actually the domain of the S protein that interacts directly with that ACE2, the protein on the outer membrane of human cells that triggers that S binds to and then triggers an infection. And so these insertions and deletions happened some period in this region, right? So they're not in SARS, and they're not in some of the strains that occur over here, but they are in many of these strains out here. And so this is when the insertions happened. And I had told you that I have evidence from my lab that when viruses get these kinds of mutations in this kind of protein, that it alters their host range. And so that was that's why I think that these probably play a key role in why SARS-CoV-2 could spread to humans and use that ACE2 receptor on humans. So we also talked about how, yes, SARS-CoV-2 is very similar to a bat strain of coronavirus, something like 96% similarity. And it has 90% similarity with a pangolin strain. So it's more closely related to this bat strain, except if you look at one small region of the SARS-CoV-2 genome, if you hone in on that spike protein, that S, there's a few amino acids that look much more similar or are identical to the stretch of the peptides in pangolin compared to the one that are in the bat. And so what we had talked about before is that this looks like a case where there's been recombination and a small segment of this RNA has been recombined with this RNA to give you something that looks like this. And it has been shown that these amino acid changes that are more like the pangolin actually give it increased ability to use human ACE2. So I think it's a combination of these insertions and deletions that happened here, and then this recombination that happened here. And it could have also been other single nucleotide substitutions and other changes in the virus that enhance its ability to spread into humans. And now we see the consequence of that. The question also is now going into the future, that's sort of the history getting us to SARS-CoV-2. Now looking to the future, is this virus continuing to evolve? And yes, of course it is. Although the nice thing is that we know that this virus has a lower mutation rate than most RNA viruses, and so it's actually evolving at a slower rate. One of the things that we talked about is whether or not it's actually adapting to humans. This is still in question, but there's some preliminary evidence that the S protein is actually adapting, and specifically in the receptor binding domain of this protein, this region here, it's accumulating mutations that are non-synonymous mutations and likely adaptive, and it's influencing, these mutations are influencing its ability to bind ACE2. This is not direct data from the lab, but this is bioinformatic data that strongly suggests that these mutations are increasing its ability to bind. But of course, we have to still do the test. But it does seem certainly that even though it has a slower rate of evolution than most RNA viruses, it is evolving and it probably is adapting to spread in humans. Whether those adaptations hurt us or make the virus more benign is still in question. And so I think we just need to learn how it's changing in order to understand not just what the original strain acted like, but what the modern strains are doing. It'll help us combat it. Okay, we have one effective therapy so far, remdesivir, that has been approved for emergency use in the United States. And since I gave the lecture on remdesivir, there's actually been a study released a little bit more on how remdesivir interferes with that RNA polymerase that we talked about before. And before I had said that it has a structure that's similar to ATP, and it inserts itself into the polymerase where ATP would, and it interferes with the construction of mRNA strands. And certainly that has been confirmed by this study, but this study goes into a lot more detail in case you want to check it out. And remember, remdesivir works today, but will it work tomorrow? There are studies out there looking at resistance mutations to a remdesivir-like molecule, but not in SARS-CoV-2. This is before SARS-CoV-2 was even out there. This is in a model coronavirus that I think infects mice, if I remember correctly. But what they found is that there are 2 mutations in the polymerase of this other coronavirus that interferes with the drug's usefulness and interferes with the drug's ability to inhibit the virus. It doesn't completely destroy the drug's effect, but it does confer some level of resistance just through these 2 mutations. And these 2 mutations rose very quickly during their lab experiments. And so it suggests that they could arise in nature. These 2 mutations are occurring at this site and this site in the polymerase. So this is a peptide strand, and it's from different, what they've done is they've aligned different homologs, so different versions of the same gene in different coronaviruses. And the coronaviruses are, there's a huge diversity of them. But what they find is that no matter what coronavirus they look at, there's always an F here and there's always a V here. So this F corresponds to that F and this V corresponds to that V. Well, in the lab experiments, that V changes to an L and that F changes to an L. And so this has 2 important points we can make from this observation. One is that, even though this study was done on a different coronavirus, these are conserved locations in all coronaviruses. And so it suggests that if SARS-CoV-2, since it also has this amino acid, or it likely does, then changing it might be able to confer resistance to remdesivir. But it also has, there's a good side to that. So that's the bad side, but there's a flip side. And it suggests that if these sites are really well conserved, we don't see any amino acid variation at these positions in the protein. That suggests that the way that these amino acids work has a big effect on how the polymerase works. And so if they mutate it, it's going to have this huge pleiotropic cost. And so it's going to handicap the virus. And so there's some idea that, well, maybe they can evolve resistance, but maybe that resistance will be obviously really bad for the virus. And so it might not be able to spread as well if it has these resistance mutations. Of course, that good thing has the caveat that we know that compensatory mutations can evolve in proteins, and nobody has studied compensatory mutations to these mutations yet, but I bet that they're available, and I bet that the virus could evolve them. So I think we do have to, as we start to use remdesivir, be very careful not to select for resistance mutations, or if we do, to isolate people that have the virus, that have the resistance mutations, so that that virus doesn't spread, rendering our therapy useless. So this is just the experimental data that supports the observation that these mutations are for resistance. Remember this, how to read this plot, and that we were calculating the EC50 from these kinds of plots. And we can see that the EC50 is higher concentration is used to prohibit the growth of the virus. So remember what EC50 is, CC50, and SI. So this is inhibiting 50% growth inhibition of the virus. This is 50% toxicity on the cells in the cell culture. You want this to be high. You want this to be really low. And so you can look at this selectivity index, which is just the ratio of this to this. So if this is really high, so it takes a huge dose of drug to really damage the cells, and this is low, it takes a very low dose of drug to inhibit the virus, then this SI index is going to be really high. And that's an indication that that's a likely drug that will work without harming the patients. And so that's very good. Okay, so remember how to look at those graphs and calculate those different things, and remember that equation. Thank you, guys. We're halfway through, and the next half is going to be much more applied and focused on actual diseases and not just these sort of concepts and understanding evolution.